So in the second podcast, we're going to look in more detail at mucus. So what is mucus? What is this protective lining? And we're going to look at the parietal cell. So mucus is composed of a network of proteins that have long sugar chains attached. So there are 19 different types of mucin proteins encoded by the human genome. Some of these mucin proteins are attached to the surface of the epithelial cell. Other mucins are secreted, and they are cross-linked together via disulfide bonds. So those are mediated via the side chains of cysteine residues. So these mucin proteins form a protein network. Attached to these mucin proteins are long sugar chains, and the characteristic of these sugar chains is that they retain water, and so they prevent the movement of water and cause an increase in the local viscosity. And so this is the gel-like layer that is located at the surface of the surface epithelial cells in the stomach. So in addition to these mucin proteins, the epithelial cells also secrete bicarbonate. And so this bicarbonate ensures that the pH at the surface of these epithelial cells is neutral. And these bicarbonate ions slowly diffuse through this gel. And as a result, the further you go away from the surface of the epithelial cell, the lower the pH until it is eventually identical to the inside of the stomach. So how are these sugar chains attached to these mucin proteins? Well, it occurs through a process called glycosylation. So glycosylation is a post-translational modification, so it occurs after the synthesis of the amino acid chain. And it should be noted that it's a very common modification of membrane and secreted proteins. It's really very common. There are two types of glycosylation. So there's N-glycosylation. So here's an example of N-acetylglucosamine linked to an asparagine. So it's linked to the amino group of an asparagine. That's why it's called N-glycosylation. And this takes place mainly in the endoplasmic reticulum. And the other type of glycosylation is so-called O-glycosylation, which is glycosylation of the hydroxyl group of serine or threonine. Hence it's called O-glycosylation. And this mainly occurs in the Golgi complex. After the addition of the first sugar moiety, the subsequent addition of simple sugars can result in these long and complex glycosylation patterns. So each addition is facilitated by specific enzymes. And it should be noted that these glycosylation patterns can be very distinct between individuals. So apart from a specific role that these sugar chains have, for instance in the mucus, these complex glycosylation patterns have an additional medical relevance. And that is because the immune system really uses these glycosylation patterns to distinguish self from foreign material. So when a donor and recipient are matched, for instance, for a blood transfusion or organ transplant, then this matching is really largely based on matching glycosylation patterns in the donor and recipient. Let's now look at these glycosylated membrane and secreted proteins and find out how they arrive at the correct location. So for instance, how do these mucin proteins, how are they secreted or attached to the membrane? So first the genes are transcribed in the nucleus, and the RNA is transported to the rough endoplasmic reticulum, where the mRNA is translated, so the polypeptide chain is formed. And the endoplasmic reticulum is also the site where N-linked glycosylation takes place. So the inside of the endoplasmic reticulum will contain the proteins that are going to be secreted, and the membrane proteins will be inserted in the membrane of the endoplasmic reticulum. So these proteins are transported via vesicles to the Golgi complex, and here in the Golgi complex further glycosylation takes place, and O-linked glycosylation occurs as well. So again, the inside of the Golgi complex contains the proteins that will be secreted, and the membrane proteins will be inserted in the membrane of the Golgi complex. So after the glycosylation is fully complete, the proteins will be transported to the apical membrane via vesicles, and after fusion of these vesicles with the plasma membrane, the inside of the vesicles will be secreted, and the membrane proteins will be inserted in the apical membrane of the cell. We've now looked in quite some detail at the mucous surface cell of the stomach and how it protects the underlying tissue from the gastric acid. Let's now focus on the parietal cell, the cell that secretes gastric acid, and it is located in the gastric pit. So here's a schematic diagram of the parietal cell. So I've indicated the receptors, the three receptors that send signals and stimulate the parietal cell, and these receptors are located at the basolateral membrane, and on the apical membrane is the actual proton pump that is involved in the secretion of the protons. Before I go into more detail about how exactly hydrochloric acid is secreted, I'd just like to focus on the dramatic changes that a parietal cell undergoes when these three different receptors receive signals and stimulate acid secretion. It should be noted that the parietal cell can really undergo quite dramatic changes when it's stimulated to secrete hydrochloric acid. So for instance, here on the left is an actively secreted parietal cell. So this is when the H2, the M3 and the CCK2 receptors are all activated, and this actively secreting cell is characterized by a large surface area of the apical membrane, so all these protrusions are formed, and conversely there is only a very small number relatively of internal vesicles, and so the proton pump is really located in this case on these protrusions of the apical membrane. So by contrast, this is a schematic of an inactive resting parietal cell. So the changes are quite dramatic. So the surface area of the apical membrane is much reduced, and conversely the number of internal vesicles is really very high, and in this case the proton pump is located on these vesicles, these intracellular vesicles, and the proton pump is inactive. So the bottom line is that this is an example of a cell that can really undergo quite dramatic changes upon stimulation. So here again is a schematic diagram of the parietal cell. This time with all the main players that are located at either the apical or basolateral membrane highlighted. So those include receptors and signaling molecules. So here's the H2, the CCK2 receptor, the M3 receptor, the G proteins and the adenylyl cyclase, which produces the cyclic AMP. And in addition, it contains a number of the key players are involved in transmembrane transport. So in the next two podcasts, I will really look in more detail at the various transmembrane proteins. But let's now first look at the mechanism of hydrochloric acid secretion and the flow of molecules that are involved. This is then a schematic diagram of the parietal cell. We've highlighted the movement of molecules across the membrane that leads to the secretion of hydrochloric acid across the apical membrane. So let's start here with CO2 at a basolateral membrane. So CO2 diffuses into the parietal cell across the basolateral membrane, where it is converted by an enzyme, carbonic anhydrase, to H2CO3, which is then converted into bicarbonate and a proton. The bicarbonate ion is then transported across the basolateral membrane, and this transport is coupled to the import of a chloride ion. The chloride can then be transported across the apical membrane by a transporter protein, while the proton is exchanged for a potassium ion by the proton pump, which is a process that requires ATP hydrolysis. So the balance of potassium across the apical membrane is then restored by a potassium channel, which facilitates the transport of a potassium ion from the intracellular space into the lumen. And so the net result is the secretion of hydrochloric acid by the parietal cell.